Another contributor to genetic variation is the rate of reproduction. So in higher eukaryotes, like animals and plants, mutation rates tend to be pretty low. They're not zero, but they are pretty low, about one mutation in every 100,000 genes per generation. However, in bacteria, archaea, and especially viruses that don't have that DNA polymerase proofreading that eukaryotic polymerases have, mutation rates tend to be much higher. They also have a much higher rate of reproduction. And we know from our studies of meiosis and looking at Mendelian basis of inheritance that sexual reproduction shuffles alleles that exist into new combinations. So organisms that reproduce sexually, this is actually more important. Sexual recombination is more important than mutation to produce genetic variability, heritable variability that make adaptation possible. So this brings us to what tends to be kind of a difficult conceptual item on the syllabus for us, which is the Hardy-Weinberg equation. What the Hardy-Weinberg equation is, it is a way of testing to see whether or not a population is evolving. So what it is a null model, which means if this model is being strictly adhered to, we would expect there to be no evolution occurring. If we find that there is if there are changes in the model, then the population is evolving. So we're wondering what's happening in a population, whether or not it is evolving. So what do we mean by a population? So we can define a population as a localized group of individuals that are capable of interbreeding and producing fertile offspring. Okay, so capable of interbreeding doesn't mean they have to be interbreeding, or everyone doesn't have to be breeding with everybody else, but at least there is the potential for interbreeding and producing fertile, viable offspring. Okay, so a gene pool consists of all the alleles for all loci in a population. If there is only one allele for a gene in a population, every individual in that population is homozygous for that allele, we say that locus has become fixed. A locus is fixed if all individuals in a population are homozygous for the same allele. Okay? So we can calculate allele frequency within a population, or how many copies of an allele, what proportion of individuals have a particular allele within a population. So for diploid organisms, which are all animals, and for the most part, most plants are found in the diploid state, the total number of alleles at a locus is the same as the total number of individuals times 2. So for any particular gene, for any locus, you are carrying around 2 copies of that particular locus. Okay? So the total number of dominant alleles at a locus is 2 alleles for each homozygote, plus 1 allele for each heterozygous, and the same is true for recessive alleles. So if someone is homozygous recessive, they are carrying 2 copies of the recessive allele, and every homozygote or heterozygote is carrying 1 copy of the recessive allele. So usually refer to a case where we've only got 2 alleles, homozygous, a dominant and recessive. Refer to those alleles as P and Q. That's what we used to refer to their allelic frequencies. What is a frequency? A frequency is basically another way of saying what percentage of all of the alleles in a population are P and what proportion are Q. So if we're talking about percentages, they're always going to add up to 100%. So the frequency of all alleles in a population for a particular locus are always going to add up to 1, which means 100%. Think of that as 100%. So P plus Q is always going to equal 1, where P and Q are used to represent allelic frequencies. How many big B's, how many little b's in a population, just as an example. So let's say we've got a population of wildflowers that is incompletely dominant, just like our snapdragon example. We use this example of incomplete dominance because it makes it a lot easier for us to figure out where all the alleles are. Okay, so we've got 320 red-flowered plants, 160 pink-flowered plants, 20 white-flowered plants. And we know the genetic basis for all of these phenotypes, these appearances. Okay, so we need to calculate how many copies of each allele. Okay, so for these 320 red-flowered plants, which have 2 copies of C superscript R, it's going to be 320 times 2, because they each have 2 copies, plus these 160 pink-flowered plants have 1 copy of this allele, so we only add them 1 time, so we see 320 times 2 plus 160 equals 800 copies of that allele in this population. Okay? Do the same thing for the white flowers. We say, all right, there's 20 white flowered individuals. So we know they've got 2 copies of this C superscript W allele. So that's where we get 20 times 2. Each of these pink flowered plants has 1 copy of that CW allele. Which means we have 160 of those. We add that all up. We get 200. So now, if we add those up, we know the frequency of this C superscript R allele is 800 over the total number of individuals is 0.8. And the frequency of the CW allele is 200 divided by 1,000, or 0.2. All right, so 80% of the alleles in this population are C superscript R, 20% are C superscript W. The sum of the alleles is always going to equal 100%. You're always going to have to account for 100% of the alleles in a population. So, the Hardy-Weinberg principle describes a population that is not evolving. It is not evolving. If the population is not meeting the criteria of the Hardy-Weinberg principle, then the population is evolving. So, what the Hardy-Weinberg principle states is that if certain conditions are met, the frequency of the alleles and the frequency of the genotypes in a population will not change. They will remain constant from one generation to the next. So we will see no microevolution, no change in allele frequency or in genotype frequency. So in a given population where gametes contribute to the next generation randomly, that is, say, where we have random mating, allele frequencies will not change, and Mendelian inheritance is going to preserve genetic variation within a population. So if we took our sample population, we know the frequency of our C big R allele and the frequency of our C W allele, 80% and 20%. That's our alleles in the population. So for each egg and each sperm, there's an 80% chance that it's going to be red, a 20% chance it's going to have the white allele. Same for the sperm. So Hardy-Weinberg equilibrium describes constant frequency of alleles in a gene pool. So if we had a population of 500 wildflowers and 100 alleles, where these are our frequencies, we can figure what we can calculate what the genotype frequency will be. So the number of CR is going to be the same as the value of P squared, or 0.8 squared is going to give us 0.64. The value of CW is going to be P times Q times 2. We'll get to why that is why that's 2 times in a minute. So that would be 2 times 0.8 times 0.2 is going to be 0.32, 32% heterozygous. The frequency of CW is going to be Q squared, or 0.2 squared is going to be 0.04, 4% white. So the frequency of these genotypes can be confirmed by using a Punnett square. All right, so why do we have P squared is the value for homozygous CR? Because it is 0.8 times 0.8, which gives us 0.64. Y is Q squared. Y is CW Q squared, because it is 0.2 times 0.2, or Q squared. Q times Q gives you CW. And why do we get 2 PQ? Because just like in a Punnett square, we've got two squares that, in a Punnett square, we've got two squares that are CR-CW. So there's one where the CW comes from the sperm and CR comes from the egg, one where CW comes from the egg and CR comes from the sperm. So two squares that give us P times Q. That's why we have 2 times PQ. And we multiply out the frequencies. We get those frequencies adding up to 100%, or 1. So 80% likelihood of getting a red allele for sperm, 80% likelihood of getting an R allele for the egg, means that there's going to be a 64% chance of getting a red flowered plant. Using those same figures, we can figure it's about 16% chance of getting a pink flowered plant where the CW allele comes from the sperm. There's a 16% chance of getting a pink flowered plant where the CW allele comes from the egg. And the chances of getting a CW allele from both sperm and egg is going to be 20% times 20%, or 4%. So if P and Q represent the frequencies of the only two alleles in a population at a locus, then the proportion that are homozygous dominant is going to be P squared, homozygous recessive are going to be Q squared, and heterozygous is going to be 2 times P times Q. However, the only time we're going to see that a population stays in Hardy-Weinberg equilibrium is if 5 conditions are met. So these 5 conditions, very important. If any of these 5 conditions are violated, the population is going to change in allele frequency or in genotype frequency from one generation to the next.